introduce. Okay, so I will first introduce Dr. Fujinaga. It is my great pleasure to introduce our speaker today, Dr. Fujinaga.
Thank you.
Dr. Fujinaga is an associate professor at UCSF. He got his PhD in the late 90s at Hokkaido University in Japan and did his postdoc under Professor Matija Peterlin at UCSF. After working at Case Western and University of Michigan, he went back and settled at UCSF in 2010. Dr. Fujinaga's main research interest is biochemical analysis of transcription regulation in immune cells. Particularly, he is an expert on a critically important cellular factor called Tefb. Over the years, he published numerous studies on the structure, function, and regulation of Tefb. As other labs verified and built upon his work, it has become clear that his discoveries are not only important in the field of HIV, but also important for us to understand the cellular immune functions. So I can't wait to listen to today's seminar entitled Tefb Turnover, HIV Latency, and T-cell Dysfunction. So during the seminar, if anyone has any question, please raise your hand. We encourage interaction, or you can leave the question in the chat. And so please join me in welcoming Dr. Fujinaga.
Thank you. Thank you very much. So I should.
You just stop sharing.
Yeah. So before I was able to share my screen, but it somehow disappeared.
Oh, just quit sharing. Maybe you can share again.
Sorry. The share screen.
Yeah.
Basic. Desktop. Okay. Good. Desktop. Okay. Sorry. Share. Okay.
Yeah, I see your screen.
Oh, okay. Good. Okay.
Perfect.
Thank you. Okay. Thank you very much. Can you see the? Okay. Good. Okay. Thank you very much for Jihan for the wonderful introduction, and thank you very much for all of the organizers for giving me a chance to present about our recent studies about Tefb turnover, HIV latency, and T-cell dysfunction. So just remember that I gave the talk at Gladstone like 2 years ago almost, and with very similar titles. But we actually had a very good progress since 2 years, and we have more interesting stuff to talk about. We have 2 papers published, so hopefully we have a lot of interesting things. And again, so I've been, as Jihan said, I've been working with Matija for like since 2010, and we worked together to run the laboratory until basically Matija retired or retiring. So exactly, I don't remember the official date, but I think last time I had was December 31, might be a little bit more. But after Matija's retirement, I'm actually moving to the group of Nevan Krogan's, Dr. Nevan Krogan's HARC, and particularly the Tefb research group led by Dr. Frankel. And I'm in the process of moving from Parnassus campus to Dr. Frankel's lab in Genentech Hall. So hopefully, actually this Thursday is a big moving day. So after basically February, I will be your neighbor of Gladstone and the Genentech people. So I like to have hopefully more interactions in person with many people as well. Okay, so let's start. So basically last almost 20 years, after leaving Matija's lab and come back to Matija's lab, we've been working on almost obsessively working on these P-B factors. P-B stands for positive transcription elongation factor B, which is a dimer between CDK9 and cyclin T, particularly cyclin T1. The main reason is because this is a master switch between transcription pausing and elongation. So my talk today, so again, anytime if you have any questions, please interrupt me in a way that I can notice. So maybe just kind of showing up or something. But basically, I'd like to first talk about why we are so much interested in P-B and why we think P-B is so important. And then we talk about recent studies. Basically, the project that we've been working on for the last 2 years about cyclin T1 protein regulation, what we call P-B turnover in cells, which is mostly done by the former postdoc Fang Huang from Wuhan. He finished 2 papers published and went back to Wuhan. And right now he's trying to set up his own group right now. And based on that, I'd like to actually introduce some of our current and future projects about how to use this knowledge for HIV and other research. Okay, so our understanding about eukaryotic cellular gene transcription is quite changed since 2010. And now we kind of know that almost all gene promoters in cells, no matter which kind of cellular status, most of the promoters, RNA polymerase II, is already engaged and basically ready to transcribe or started transcription, initiate transcription. But usually everything is paused by mostly by the factor of NTAF or negative transcription elongation factor. And so most of the genes are like this. And then when P-B is recruited to the POL2, the gene started transcribing. So therefore the P-B, and when P-B is recruited to POL2, it phosphorylates the CTD region of polymerase II, as well as the negative transcription factors, which is removed after P-B is recruited. So this is a main on-off switch of transcription pausing and transcription, productive transcriptions. And recently, Patrick Kramer's group actually published 2 very important Nature papers on the structure of this paused POL2 and elongating POL2. Both are resolved by cryo-EM studies, which is very, very informative, big breakthrough in transcription. So then the big question is, one of the big questions is how P-B is recruited to POL2 whenever it's required. So this is one of our big project, which I'm not talking today, but briefly going through. For example, POL2, P-B is recruited through enhancer, upstream or downstream enhancer, through DNA looping and some other DNA sequence-specific enhancer binding proteins, including NF-kappa B, STAT6, MEF2, c-MYC, ER, and other stuff. Also, from HIV, we knew that P-B could be directly recruited through RNA. And also, P-B can also be associated with promoters through other factors such as C2TNA, other stuff. And also, P-B is epigenetically regulating the transcription through the interaction between BRD4, particularly. And at last but not least, there are some factors associating with POL2 sequencing in the manner sequence independent, such as PAF1 for RNA2 associated factors or super elongation factors, which will come later a little bit. So So P-TAB is associated with many diseases and conditions, and particularly when the P-TAB activity is abnormally upregulated, that's associated with cardiac hypertrophy, for example, and various different types of cancers, particularly through the regulatory factors like ER, and some inflammation and autoimmune disease. Also, P-TAB is particularly up. And also, there's a situation that P-TAB activity is abnormally down. This is where HIV is going latency in the resting or quiescent T cells, or some example of T cell responsiveness, such as exhaustion and energy, which we are going to expand our study into from HIV as well. And other viral latent infection infected cells, such as HTLV1 or other DNA virus, which use PO2 and P-TAB as a cellular factor for transcription of viruses. And also we have some situation that genetic rearrangement force P-TAB to the specific loci, which has happened in multiple lineage leukemia. And this led to the discovery of this SEC mutant by the SEC complex by Shiratifat group. So basically, because of P-TAB activities closely associated with many diseases, main sort of motivation for us to study P-TAB is actually, if we can manipulate P-TAB, that will be clinically beneficial for different types of disease. So this is why we think P-TAB is important. And particularly P-TAB is important for HIV and AIDS, as many people know. The biggest reason is basically P-TAB binds to this HIV TAT protein as well as HIV tRNA and make a transcription elongation complex for HIV. So without that, HIV doesn't go beyond that. Transcription doesn't go beyond that. Therefore, HIV doesn't make any productive infection. And also, P-TAB is known to be recruited through NF-kappa B P65, P50 complex by direct binding. So NF-kappa B is also very important for HIV transcription. So basically, two major factors critical for HIV transcription. One is TAT-TAR, the other one is NF-kappa B. Both required P-TAB. So therefore, P-TAB is particularly important for HIV transcription and latency. So everybody knows almost the biggest problem of HIV is the latency. So as we know, so green is sort of immune functions by counting CD4 positive T cells. The black line is HIV viral load in peripheral blood. So as we all know that we have a good drug for highly active antiretrovirus therapy that can reduce the HIV viral load to the undetectable level. And as long as the patient takes medicine, they're fine, no virus coming out. But once they remove HARC at any occasion, the virus suddenly comes up and rebound, and that causes severe viremia and AIDS. So therefore, the biggest problem of HIV is latency. And we have basically two schools of thought in terms of HIV functional cure targeting latency. One is the so-called shock and kill approach, which somehow try to prevent latency by keep activating HIV so that HIV is removed by immune function and by its virus, its own cytopathic effect. And also we have another way called block and lock strategy, which basically try to keep the virus load down all the time, even without HARC. So for both approaches, transcription of HIV is a key step Therefore, again, manipulating P2P activity, which is critical factor for HIV transcription, either up or down will be beneficial for both strategies of shock and kill and block and lock. So therefore, that is actually our biggest sort of justification of P2P study in the context of HIV. So so P2P activity, P2P basically is regulated in cells by two main mechanisms. One is how P2P is recruited to post-RNA polymerase 2. So again, so there are a lot of different ways to recruit P2P to POL2, and we know that not all genes are activated at the same time. So there's a selection between genes to be activated and genes not to be activated. So there should be a lot of different types of mechanism of how P2P is selectively recruited to POL2, maybe through other factors, protein factors, or maybe some DNA or RNA sequencing specificity, and or these kind of things are also should be tightly regulated by some environmental or developmental cues, such as signaling, epigenetic mechanisms, and so on. And also there's a big mechanisms of regulating P2P functions in cells, not just recruitment. One is also these functions are recruited mainly, there are two big functions, regulation of functions of P2P. One is the kinase activity itself, because kinase activity is important for critical for the transcription activity. And also P2P is regulated tightly by protein levels and localization, which I'm trying to talk in the next section. So, and this regulation is also tightly regulated by environmental and developmental cues, and also associated with disease and conditions to manipulate and then how to manipulate P2P is very therapeutically important. So the function P2P function regulation number one is so far the most well and extensively studied mechanism called 7SK SNAP regulation. So most of the cells, the replicating cells, P2P is expressed in a high level, but most P2P molecules are incorporated in 7SK SNAP complex, which is 7SK RNA, hexim1, LAP7, MEPC, and other proteins all make a huge protein complex. And in this complex, CDK9 kinase activity is inhibited by this hexim1 protein. Therefore, CDK9 is enzymatically inactive. Therefore, it doesn't activate transcription. And many cells stimulation, environmental, developmental cues and so on, release P2P from this complex and activate it. And that activated P2P is the one to be eventually recruited to RNA polymerase 2 to start transcription elongation. And after the job is done, cyclin T1 P2P complex is going back to 7SK complex and kind of going back to the inactive state and wait for the next signals to come. So this is our biggest sort of well studies of the P2P regulation in replicating cells. And I would say most of the so-called latency reversing agent to be used in the shock and kill approach are tackling this step, particularly to release P2P from 7SK SNRNP. But there's another P2P regulation, particularly in resting T cells, where basically most of the HIV latent reservoir is found. So in these cells don't replicate, and in these cells, the biggest regulation of P2P is cyclin T1 protein going down. So this is one of the typical experiment to establish resting T cells. So first you take blood cells and purified T cells. First you stimulate by CD3, CD28, FPS, IO2, etc. to expand. And usually after that, you for 24 hours or something, you remove everything gradually to the almost zero, just to not to kill the cells. Then cells goes to the resting state, and in that stage, the cyclin T1 proteins go back to almost undetectable level. But what we observed was when in this condition, when the cells are treated with bortezomib, which is a clinically approved proteasome inhibitors, one of the clinically approved proteasome inhibitors, the increased cyclin T1 protein to the level equivalent to cyclin T1 in the stimulated cells. That means, and also it's importantly, cyclin T1 messenger RNA level didn't change during this treatment or stimulation or anything. So basically, that means that in resting T cells, the cyclin T1 proteins are rapidly, severely downregulated through the proteasome degradation. And there's also other data showing that the shRNA is involved, but the fact that bortezomib almost rescued the cyclin T1 level to the level of stimulated cells implies that the proteasome degradation is the major mechanism of cyclin T1 downregulation in resting T cells. And that is very important for studying HIV latency, because when you study like Jarkat cells or any replicating latent model, this doesn't happen. So then whatever strategy coming up with the study with the replicating cells might not be working in the resting T cells, which is actually the latent reservoir can be found. So therefore, I think it's important to study this mechanism, particularly in resting T cells. And interestingly, we found that similar cyclin T1 downregulation can be seen in some of the models of T cell unresponsiveness. So unresponsive T cells are different types of there are different types of unresponsive T cells in body. Some of them are like T cell allergic T cells, and some of them are exhausted T cells. And particularly this T cell exhaustion is very important, which is frequently seen in patients with chronic viral infection and cancer, where the T cell is constantly chronically stimulated by the same antigen over and over. And in these cells, T cells do not respond to the TCR stimulation anymore, and therefore it reduces greatly reduces the efficacy of cancer immunotherapy or CAR T cell therapy, because they need active T cell actively responding to T cell receptor. So therefore, it's very important to study, and we did a couple of different in vitro model of T cell unresponsiveness, and one in vivo model of T cell allergy. And I don't go into the detail, but in any models, we see cyclin T1 protein level going down, like the one in resting T cells. And this protein, again, messenger RNA didn't change. The protein level is rescued by bortezomib, so it's kind of similar. Looks like a similar mechanism is happening in at least some of the unresponsive T cell models. So now we add basically one more layer of cyclin P2P regulation, particularly in resting and dysfunctional T cells, where cyclin T1 level is down mainly by cyclin T1 degradation. And we know already that CDK9 protein is stabilized when cyclin T1 is not there by HSP 70 and 90, which is a Chanzu's paper 2001, 2000 JBC paper demonstrated that. So therefore, there's some kind of mechanism in cyclin T1, particularly degrading cyclin T1 in these resting T cells. So so now we concentrate on this beta P or cyclin T1 turnover because this severe downregulation of cyclin T1 proteins, in particular in resting T cells where the HIV latent reservoir is found, could be a very major cause of HIV latency because, as I mentioned, without beta B, the transcription of HIV doesn't go beyond the TAR, so it doesn't make any productive infection. So we have to have cyclin T1 beta B for the HIV replication. And also, severe downregulation of cyclin T1 protein in responsive T cells may be also a major cause of T cell allergy or exhaustion. So because it doesn't, one of the reasons of the T cell unresponsiveness is it doesn't transcribe any T cell receptor dependent genes. So from that, we hypothesize that we have a question. Blotowski, okay, yeah.
Are you looking at the question?
Okay, yeah, I see Ursula's question right now. Yeah.
I think we can maybe discuss this at the end.
Okay. Yeah. Actually, I didn't. Yeah. Okay. Sorry. All right. So I just saw the. Okay. So that we hypothesize from that by increasing cyclin T1 proteins in these cells may reverse prevent HIV latency as well as T cell allergy or exhaustion. So so Fang trying to understand this mechanism of cyclin T1 degradation. So she started basically looking at old literature and found actually interestingly old paper. When I was in Case Western, I have a before rejoining Matija's lab, we published one paper in JGB showing that three highly conserved threonine residues in cyclin box in cyclin T1 is important for P2P function. And when these three threonines are mutated to alanine, this P2P becomes dominant negative on HIV transcription. But we haven't really, we hadn't done much mechanistic biochemical studies in that paper. So he took this paper and sort of start studying about this mutant, what he called T3A mutant. And first he expressed this mutant in cells, in 23 T cells, and he found that in steady state, this protein expressed very poorly. But again, when cells are treated with bortezomib, the protein levels came back to the normal level. So again, this mutant is very short-lived protein with rapidly degraded. The one similar to the cyclin T1 in resting T cells. And he did a pulse chase experiment showing that actually the short half-life of T3A mutant is much shorter than the wild type. And also, basically my presentation today is most of the data are IP Western. So to make easier to follow the IP Western, so I just put the diagram of what is IP and what is Western, Western in like here. Anyway, so he did the core IP experiment to look at cyclin T1 CDK9 interaction, and he found that T3A mutant doesn't bind to CDK9. Even the protein level is rescued by bortezomib. There's no interaction. So basically, there's a connection between no interaction between cyclin T1 CDK9 and rapid degradation in cyclin T1. So basically, we hypothesize that the cyclin T1, which doesn't bind to CDK9, is rapidly degraded in cells. So to confirm, he did the ubiquitination assay comparing wild type and this T3A mutant, and found that T3A mutant is actually heavily ubiquitinated in cells. The next, since threonine residues is involved, he suspected that there's a phosphorylation going on. So he did. He treated the cells. This time, it's primarily CD4 positive cells, where cyclin D1 is degraded heavily. He treated the cells with oxalic acid to inhibit phosphatase, and did Western and IP also in the presence of bortezomib. And he found that after high concentration, 1 micromolar concentration of oxalic acid, there's a double band, and also there's a more interaction between CDK9 and cyclin D1 detected. And also the bound cyclin D1 is heavily phosphorylated. And since oxalic acid inhibit PP2A in the lower concentration, 5 nanomolar, and it inhibits both PP1 and PP2A in higher concentration, 1 micromolar. This phenotype is only seen with higher concentration of oxalic acid. Basically, we conclude that protein phosphatase, particularly PP1, is involved in cyclin D1 dephosphorylation. And if you inhibit dephosphorylation, cyclin D1 is phosphorylated, and it binds to CDK9 well in resting T cells. And now he went back to mutant to map the phosphorylation to confirm that actually threonine 143, 149, and O145 is involved in phosphorylation. So we already, this actually some other experiment excluded T155. So we went to concentrate on T143 and 149. So this is the result of IP Western with phosphothreonine antibodies. So once we compare wild type and 143, 149 mutant, basically the phosphorylated cyclin D1 is heavily diminished in this mutant, showing that again T143, 149 is phosphorylated in cells. And then we did a little bit of a motif search, and we found that this 143, 149 is having a very good match of the PKC phosphorylation site. So we did founded treatment of cells with kinase inhibitor staurosporin to start with. And staurosporin is known as a PKC inhibitor, but also it's a bit of promiscuous inhibitors. So after that, we went on to more specific kinase PKC inhibitors and get the eventually same result. But we just show only this time with staurosporin. But when the cells are treated with staurosporin, the steady state protein level of cyclin D1 went down very, very quickly. And also this is rescued by bortezomib. And also co-IP experiments show that when cells are treated with staurosporin, the interaction between CDK9 and cyclin D1 is also diminished. Again, cyclin D1 CDK9 interaction and cyclin D1 degradation is tightly connected. So we got some dominant negative mutant of PKC showing that overexpression of dominant negative PKC reduce the cyclin D1 by co-IP. And also dominant negative PKC reduce the interaction between CDK9 and cyclin D1. Again, showing that PKC phosphorylation of cyclin D1 is important for interaction between CDK9 and cyclin D1. And finally, we did in vitro kinase assay to more specifically studying the PKC phosphorylation on T143, 149. So the experiment was we did wild type T140, 149 WA mutant, express IP and mixed with purified recombinant active PKC and did Western by P threonine. So as you can see, wild type is phosphorylated by PKC, but mutant is not. So again, showing that T143, 149 is phosphorylated by PKC. So from this, we conclude that phosphorylation of T143, 149 in cyclin T1 by PKC promotes Tefb assembly and cyclin T1 stabilization, and dephosphorylation of cyclin T1 by very likely PP1 promotes Tefb disassembly, releasing cyclin T1 free cyclin T1, and that is further degraded. And then next question is what is victim machinery, particularly E3 ligase that mediates cyclin T1 degradation. And so we, before starting, there are several studies, particularly the cyclin T1 mass spec studies, Tefb mass spec studies, show that there is at least one E3 ligase associated with Tefb, which is FBXO11 done by Andy Rice group. So we thought that the FBXO11 is a good candidate. We CRISPR'd it, we siRNA'd it, shRNA'd it, and then nothing happened. So we conclude that FBXO11 is not involved. And other T cell specific E3 ligase, we tried GRAIL and other E3 ligase, particularly siRNA shRNA experiment, and we didn't see anything. So these E3s are not involved. And then Fang focused on this SIR1 E3 ligase because, again, Chanzu's group before published that SIR1 degrades ELL2 subunit of superelongation complex, particularly ELL2 not in a complex is a good candidate, the substrate of SIR1. And since superelongation complex is functionally very associated with Tefb, he hypothesized that CR1 also is involved in cyclin T1 degradation. So we got some construct of CR1 Cy2 wild type and dominant negative from the Brunei's group in San Diego, and we tested first with overexpression experiment. So this is cyclin T1, T3A mutant again, steady state expression, it's much less than wild type. And T3 wild type CR1 CR2 overexpression even decreased cyclin T1, particularly the wild mutant, not a little bit of the wild type as well. And interestingly, the mutant, dominant negative side mutant rescued cyclin T1, wild type cyclin T1 mutant to the level of wild type actually, but not much of the wild type. So the dominant negative mutant actually rescued T3A mutant. And then we also continue collaboration with Ronai's group, who has two categories of specific inhibitors of SIR1 Cy2. One is this naturally occurring SIR1 Cy2 inhibitory peptide called PHYL, and the other one is two kinds of small molecule SIR1 Cy2 inhibitors, RSL 24 and 26. Both are generously provided by the Brunei's lab. We tested overexpression of PHYL again. It's rescued the cyclin T1, particularly the mutant, to the level rescued by bortezomib. And also this RSL 424, 96 also rescue cyclin T1 proteins. And And finally, shRNA knockdown of CR2 also rescued cyclin T1 level, particularly mutant ones. So therefore, CR1, CR2 are the most plausible candidate for E3 ligase. And finally, we tested in resting T cells. Again, we made a resting T cell experiment. Day 7, cyclin T1 is down. Both Cy1, Cy2 is expressed highly. And in this condition, we overexpressed PHYL, and that rescues cyclin T1 level in resting T cells. So supporting the hypothesis that Cy1, Cy2 are involved in the cyclin T1 degradation in resting T cells. So now we have identified several factors involved in this cyclin T1 degradation in resting T cells. So between resting and proliferation, so PKC phosphorate cyclin T1, stabilize Tefb complex, and stabilize cyclin T1 in proliferating cells. And in resting and probably in the dysfunctional cells, PP1 activities overcomes somehow the phosphorate cyclin T1, and that release dissociates cyclin T1 from CDK9, and free cyclin T1 become a substrate of Cy1, Cy2, and degrade rapidly while CDK9 is stabilized by HSPs. This reminds basically is the regulation of cell cycle cycling CDKs like cyclin D CDK2 complex, which is also regulated by protein degradation. So we basically propose that the similar mechanisms happen between cell cycle cycling CDK and transcription cycling complex in a different type of situation. So now we have everything put together. In resting T cells, there's a big regulation of cyclin T1 dephosphorylation followed by cyclin T1 degradation. Therefore, P2P level is down. But the normal cells, PKC stabilizes cyclin T1 CDK9 complex. But yet this complex is inactivated by 7th and RNAP. So we need additional stimulation to activate P2P, which is eventually recruited to the PO2. So in terms of HIV latency, either reversing or maintenance, we have to consider all this mechanism of P2P. So now, based on this, we have several projects going on. Basically, three major projects going on. So first project is to rescue cyclin T1 protein in these cells and what happens. So obvious approach is to construct and express phosphomimetic cyclin T1 mutant, which should be more stable in these cells and more functional. And of course, we might inhibit Cy1, Cy2 to try to see what happens. And we tried after that, we will study HIV latency and activation, T-cell responses, and more globally RNA-seq experiment, etc. So we try. So the most obvious step, the first obvious step is to make phosphomimetic mutant is to change these phosphorylated residues of 143, 149 from T to E or D. But unfortunately, this simple T to E mutant or T to D mutant didn't rescue interaction between cyclin T1 and CDK9. It actually acts like T4349AA mutant. And we looked at a little bit of the structure of this phosphorylation site in cyclin T1 by collaboration with Andre Charlie's particularly Ignacia and Ramachandran are helping us a lot about modeling this this structure. what they suggest is basically T143 residue is exposed to the CDK9 cyclic T1 interface, and this phosphorylation can enhance the interaction by adding negative charges. But T143, on the other hand, is going exposed into the molecule, so it's involved in looks like it's involved in intracellular intramolecular interaction to stabilize the cycling box structures. And if you look at the phosphodonor of cyclin T1 molecules, there's some this one residues glutamine 1977 is the closest one. So and we hypothesize that adding E, changing T to E, just add only one negative charge to this residue is not sufficient to actually mimic phosphorylated status, which should add two negative charges. So we tried to compensate this one fewer negative charge by changing Q77 to basic amino acid like arginine by adding one positive charge. And this mutant actually worked well in terms of cyclin T1 CDK9 interaction. So that's now we have a good promising mutant, phosphomimetic mutant, which binds very well with CDK9. So what we are doing right now is to introduce cyclin T1 this triple mutant in primary CD4 positive T cells by lentiviral vector, which is now done by on-chain, one of the part-time research associate in the lab, and then sorry. Okay, I should talk with Ursula again. Okay. Okay. You know, overexpressed vitamin latent. Oops, sorry. Yeah, I will probably. We haven't done yet expressing cyclin T1 in latent cells, but we're gonna start doing experiment right now. We are doing experiment right now, and then after expressing this mutant as well as the wild type, the cells will be put into resting state, and cyclin T1 expression is measured. Other exhaustion marks, activation marks are measured. Then we challenge HIV, reporter viruses, and do latency experiment. If the latency is established, we stimulate the cells by LRAs to see if it actually works better or not. And then we do more thorough genome-wide analysis of constitutively active expressing cells, assuming that lots of genes are not lot of genes are actually activated, because even though you activate, you have more PTB, it should be incorporated in 7sk. So only works, our hypothesis it works only better only when cells are further stimulated by the latency reversing agent. So now the second project, it's also ongoing right now, is to identify cyclin T1 degradation complex. So we have identified several players for this cyclin T1 degradation in resting T cells, such as PKCPP1 and SIA1, and we will also try to identify more factors, basically by hypothesizing that from this to this, there should be some protein complex which is specifically associated with free cyclin T1 to transport this cyclin T1 into the ubiquitin machinery. So we kind of name this putative complex cyclin T1 degradation complex. So this is going to be being done by collaboration with Judd Hodges lab, who is Nevan Krogan's alumni. Now he's in Northwestern, and he's also the active member of HIV HARC, Dr. Krogan's HARC study right now. So we have an active collaboration between Judd's lab right now. So of course there are several P2P interactome experiment papers published by doing mass spec of P2P, but all studies are done by P2P, like active functional P2P of cyclin T1 CDK9, because they use 293 T cells or any cells where P2P is stably expressed. So those interactome always come up with factors like CDK9 or 7SK, BRD4, and so on. But our study, our plan is actually to particularly enrich this complex. So this was we call normal cyclin T1 interactome, but we try to do mass spec with cyclin T1 rescued by borderless, and from that we hope to enrich proteins which is involved in this pathway, which is Cy1, hopefully Cy1, Cy2, PP1, and other proteins. And this we call cyclin T1 degradation complex. And each one of them from this study could be a new therapeutic target for HIV/AIDS and T cell responsiveness, because we can manipulate this pathway by dealing with these factors. So the third project right now is to monitor cyclin T1 degradation in vivo by fluorescent based assays. So we constructed this dual color lentiviral vector, one expressing YFP cyclin T1 fusions and CFP connected by iris. And when the cyclin T1 is degraded, YFP is degraded as well, while CFP protein remains high because it's expressed independently. So, so investing T cells, CFPs used as a monitor of lentiviral transduction, and YFP is actually is going down because of the cyclin T1 is degraded. And if it's rescued by bortezomib, YFP goes up, and PMA PHA stimulation as well, YFP goes up. So this is preliminary result, but using these cells, these systems, what we call dual color reporter cyclin T1 turnover assays or dark assays. Using this system, we can monitor the rate of cyclin T1 turnover in single cell level to hopefully to identify specific T cell subtypes where HIV goes more deep latency because cyclin T1 is more severely downregulated, and use this system to for the high throughput screening, small molecule screening, to discover new and more effective latency reversing agent. And also we can use this system to do high throughput screening using the shRNA or CRISPR to identify cellular factors involved in cyclin T1 turnovers, also, which could be a good, anything coming up could be a good new therapeutic target. And also we use this similar system by using T143, 149 mutant, which is more highly degradable version of
Sorry to interrupt. We have 5 minutes.
Okay, this is actually last slide. Yeah, thank you. So this is going to be used for particularly for the shRNA CRISPR screening in cells where cyclin T1 is more stably expressed, like 23 T cells as an alternative approach. Also use the drug screening as well. So yes, this is actually the last slide. So all the studies are done in the lab, and it's Matija, and most of the biochemical experiments are done by Frank, who is now back in Wuhan, and Dan and On, current member. Actually, they are sort of in transition from Parnassus to Mission Bay as well. Both are part-time, not the full-time, but they work under their own schedule. And Daniel, Hannah, Rashad, Marie are all former postdocs who went back to basically different, either their own countries or different laboratory. They're all also involved in these studies as well. Zipingo was the long-term lab manager in Matija's lab. He contributed a lot of studies. Now he's in Mission Bay, actually, in the laboratory of Fujimori lab and Jura labs. Kind of they sort of share Zipin right now. So now I'm going to the neighbor again with Zipin. And in terms of collaborators, the T-cell energy mouse model experiment was done by Art Weiss. I'm sorry, particularly Trang, postdoc, who is now back in Vietnam. And second-year structure studies under Shari lab, particularly Ignacia and Ramachandran. Sci 1, Sci 2 is Stanford Barnum collaboration. Zabronai lab, particularly Yon May, is a postdoc. And mass spec analysis is done in Judd Hodgkins lab in Northwestern, particularly by William, who is a student of Judd. And fundings NIH and HARC, Dr. Krogan's HARC, Treadwell, and from UCSF lab supported these studies. And again, thank you very much, and Happy New Lunar Year. Thank you very much for attention.
So thank you, Dr. Fujinaga, for the wonderful research. Also, very good data. So there are three questions in the chat, and I will read the first one for everyone. Okay. So from Ursula, what is your opinion on single molecule research from Robin Borotowski's lab? They found no evidence of polymerase II pausing. Rather, they think that elongation factors simply increase percentage of successful long transcription products. Short transcripts from PAL2 are non-productive. PAL2 falls off and no pausing. And Ursula is asking your opinion.
Yeah, I haven't actually read this paper very carefully yet, so I have to actually think very carefully. But it's very generally speaking, it's very hard to reconcile some of the genome-wide studies and so on to the other studies. So I'm actually not quite sure how to answer this question. Because
yeah, I didn't want to turn into like
I know.
I heard him talk at some point, and I was kind of very surprised that he totally sort of rejected this whole idea of pausing, you know. And so I was just wondering if you had sort of an opinion on that.
Yeah, different experiment. For example, John Lee's experiment clearly said that there's a PAL2 just paused, you know, in the Drosophila study as well, and all the ChIP-seq experiment also suggests there's a pause release. So it's hard to actually,
so I guess it's maybe controversial.
So it's probably, but I should probably read the paper. Is it actually published recently? I don't remember.
I don't know. I just got this from his presentation here like half a year ago or so. So,
that's actually I was not aware of this, but I have to look at this study. It's published right now, so I'm sorry about that.
Okay.
Thank you. Second question from Udru. Has anyone tried overexpressing Tefb in latently infected resting T cells or exhausted T cells? And this is asking whether there's an effect in their cells.
You haven't actually. I don't. As Ursula said, I don't recall this. No, this is sorry, not Ursula. I don't see papers, studies like this. And we actually, we haven't done that yet with this experiment.
So are you planning to do this?
Yeah, actually, this, of course, when this mutant is expressed resting T cells, we use wild type as a control, assuming that wild type is also degraded quickly. So again, the circumstantial evidence is all the cyclin T1 downregulation is post-transcriptional. So introducing more genes, cyclin T1 genes, increasing messenger RNA of cyclin T1 would not increase cyclin T1 proteins. This is our assumption. But this is actually coming when we do the experiment with the mutant. Yeah. And then, yes, sorry. Yeah.
Thank you. So the last question is also from Ursula. So she said, I don't recall that cyclin T1 phosphorylation has been seen in any structure, any structures of recombinant Tefb. Is cyclin T1 phosphorylation in a flexible loop?
So first, I think, yeah, this is actually the reviewer's comment as well, in particularly the eLife paper we published for the phosphorylation. And basically, crystal structures done, it's basically cyclin T1 was made by bacteria. That is the reason why we don't see cyclin T1. And some proteins, some experiment with some, I think that David Price might have used the insect cells and then express cyclin T1 CDK9 differently and reconstitute. And that experiment, I don't remember how much actually they use phosphatase inhibitor and so on. So our assumption is basically those phosphorylations are easily removed by all the processes, and particularly if cyclin T1 is used expressed and purified from bacteria, phosphorylation won't be seen. So that's why we don't see phosphorylation in the structure. And also we tried actually mass spec analysis, and we don't see, but phosphor mass spec analysis miss many non-phosphorylation. So I think one of the answers is basically cyclin T1 phosphorylation on that residues are very dynamic. That's why I think sometimes it's kind of difficult to detect by structure or mass spec analysis.
Yeah, I guess my question is, I would like to know, sort of, I mean, if I understood this right, you were saying if there's no phosphorylation, then the complex is not that stable, right? Or
right.
So let's say there was no phosphorylation in the expression, but then you wouldn't have a very stable complex, right? So that would not. So then it would be hard to purify the complex, right?
Bacteria, you don't have to worry too much. Probably, and bacterial, depending on how the degradation machinery is specific to cyclin T1, because we do actually have a data show that the Cy1, Cy2 binds to cyclin T1, particularly in the absence of CDK9. So it means there's some very rather strong association between free cyclin T1 and E3 ligase. So therefore, one assumption is if you express this mutant in bacterial or something, there might not be E3 ligase which is tightly enough to bind.
No, I mean, I guess my question is the complex stable if there is no phosphorylation on the threonines in the cyclin T? The complex with CDK9, sorry.
So basically, with our model is basically cyclin T1 is dephosphorylated. It dissociates. It's from CDK9. Cyclin T1 CDK9 is dissociated, and only dissociated status cyclin T1 binds to Cy1, E3, and degraded. So therefore, once cyclin T1 is dephosphorylated, Tefb is unstabilized. Not stable.
Yeah, but so that makes then not really sense if you say, well, in the structures the cyclin T was expressed in E. coli and it's maybe not phosphorylated. Well, if that's the case, then it wouldn't be a stable complex with CDK9, and then you would not be able to purify and determine the structure, you know? So somewhere there's a little bit like a conflict.
I think, I don't know how much, for example, this complex study, do they purify as a complex, or do they actually reconstitute?
No, you put I mean, we always purify the complex from baculovirus,
Okay.
Anyway,
there might be some phosphorylation.
I think the context matters, whether you purify it from different species of cells or treat it with different conditions, buffer conditions and other conditions. I think it depends on how you do the experiment.
Yeah. So also, so I just use the word dissociation by completely. It doesn't really mean completely dissociated, because weakened might be enough, because Psi1 actually again binds to cyclin T1 very well. So that means actually if the interface between cyclin T1 and CDK9 is a little bit loosened, that might be enough for Psi1 to go into cyclin T1 and take cyclin T1 from CDK9 and bring it to the ubiquitination machinery. This is one of the explanations.
That
machinery.
I would like to continue this line of discussion, but unfortunately we are well past the time. I want to see if there's any other question from the audience. Okay, so if there's no question, so let's thank Dr. Fujinaga again for the wonderful presentation. And also, Dr. Fujinaga will have a meeting with Trini in another Zoom number. So please come over and meet with the Trini at Gladstone.
Okay, so I should remain on this. I should remain on this Zoom, or
there's another Zoom number. Please check your calendar.
I will. Thank you.
Thank you so much. See you later.
Very useful discussion. Thank you very much, Rusa.
Thank you.
Thank you.
Thank you.